Hi everyone, and welcome to our video on the link reaction and Krebs cycle. This is part of our Module 5 series on respiration, and in this particular video we're going to be having a look at three different specification references. So 5.2.2d, which is the link reaction and its site in the cell, 5.2.2e, the process and site of Krebs cycle, and 5.2.2f, the importance of coenzymes in cellular respiration. So what you can see on the specification reference is clear references made to the actual names of specific chemicals you need to know, and perhaps more importantly, the ones you don't. So if you're using a range of resources to revise, do make sure that you're doing so in conjunction with the specification so that you're not learning additional names of chemicals in these processes that you're not expected to know for your A-level course, because all that's going to do is occupy your time, which could be better spent on gaining a deeper understanding of other concepts. What we're going to do then is start off with the link reaction. Now, if you remember, at the end of our video on glycolysis, we'd ended with two molecules of pyruvate. Now, pyruvate is a 3-carbon compound, and that was produced in the cytoplasm of our cells. So the first thing that's got to happen is that pyruvate has to be transported from the cytoplasm and across the outer and inner mitochondrial membranes into the matrix. So the first thing we need to do there is to move that pyruvate from the cell cytoplasm across the outer membrane, across the inner membrane, and into the matrix of our mitochondria. And in order to do this, what we actually need is a pyruvate hydrogen ion symport. And that word symport just tells us that this is a type of transport protein which is going to be moving two or more ions or molecules in the same direction at the same time. So symport always moves together in that same direction there. The link reaction itself then is obviously, I would hope obviously, going to be taking place in the mitochondrial matrix. That's why we moved the pyruvate there after all. It wasn't just for fun. Now, when we actually get to the mitochondrial matrix, then what's going to happen is our pyruvate, so we start off with our pyruvate, remember, which is a 3-carbon compound, is going to be decarboxylated. Now, decarboxylation basically means that we're removing carbon dioxide because we're removing a carbon and it's going to form this carbon dioxide. So that is what we refer to as decarboxylation. Now, we also have dehydrogenation taking place because what we're also going to see happening is our NAD is going to form reduced NAD. And that is the process of dehydrogenation, which basically means that we're going to be removing a hydrogen from the pyruvate and then attaching it to our NAD. So what we're going to see here is we've got these two processes going on to our pyruvate. Decarboxylation, removing the carbon in order to form our carbon dioxide, and dehydrogenation, the removal of hydrogen, which attaches to NAD to form reduced NAD. Now, each of those does require a specific enzyme, and what we find is the decarboxylation is going to be pyruvate decarboxylase. So decarboxylase because it's an enzyme that is carrying out decarboxylation, and it's doing it to pyruvate. So pyruvate decarboxylase. The dehydrogenation is going to be pyruvate, because it's happening to pyruvate, dehydrogenase, the enzyme that is dehydrogenating. So what we end up with at the end is this substance called acetyl. Now, acetyl is a 2-carbon compound, so we've got our acetyl, but we're also going to be bringing in a substance called coenzyme A. So what we actually end up with is acetyl coenzyme A, or acetyl-CoA for short. So what we've got at the bottom there is just our little summary of what we've discussed in the link reaction. Start with pyruvate, decarboxylate to take it down to a 2-carbon compound, which is going to be our acetyl. We then also remove a hydrogen through that dehydrogenation, and that's attaching to our NAD to form reduced NAD. Coenzyme A comes in, attaches to our acetyl group, and that gives us our acetyl-CoA at the end. It is worth remembering that when we're talking about the link reaction, one molecule of glucose is going to have formed two pyruvates, and therefore what we're going to see happening is the link reaction will occur twice. So each pyruvate undergoes that dehydrogenation and decarboxylation. The acetyl group formed will join with the CoA to form those two acetyl-CoAs. So what that means we end up with are two reduced NAD, two carbon dioxide molecules, and two acetyl-CoA. And the reason there's two of each of them is because two pyruvates go through the link reaction. The next part of our respiration process is Krebs cycle. Now, this is taking place in the mitochondrial matrix as well, and the lovely diagram that you can see there is taking you through Krebs cycle itself. Now, we're going to look at where we're starting, our acetyl-CoA, and that was our end product from the link reaction, remember. So our acetyl coenzyme A is going to be coming in, and do notice the first thing that happens is the coenzyme A is split off from the acetyl group. And the acetyl group is then going to feed in and combine with a 4-carbon compound called oxaloacetate. So 4 carbons plus 2 carbons gives us a 6-carbon compound called citrate. What we then see is a series of intermediates, and think back to the specification reference we looked at the beginning. We don't need to know the names of any of these intermediates. We need to know our acetyl-CoA, oxaloacetate, citrate. They're the only names. You should, however, understand this part of the Krebs cycle at the bottom. It's not about knowing the names of these compounds, it's about understanding what's happening as it goes round that lower part of Krebs cycle. So, from citrate, the first thing that's going to happen is carbon dioxide will be removed, decarboxylation. We also are going to form reduced NAD, which means we have dehydrogenation taking place. That gives us a 5-carbon compound. We then remove another carbon dioxide and form more reduced NAD, so decarboxylation, dehydrogenation, to get to a 4-carbon compound. From that 4-carbon compound, we go to another 4-carbon compound, and the reason we have that conversion is we're going to generate ATP. From there, we then go from that 4-carbon compound to another, and we're going to generate some reduced FAD, so that means that more hydrogens are going to be transferred at this point, and then we're going to reduce some more NAD before we convert back to the oxaloacetate. So what we can see is we've got a few things sticking off the outside here. They're the bits that you really need to know. So you need to learn oxaloacetate, the acetyl-CoA coming in, which is split into coenzyme A, which goes elsewhere, and the acetyl group, which feeds in to join to the oxaloacetate. That forms our citrate, need to know that name, and then the rest of it, you just need to know where the carbon dioxides are, where the ATP is, and then where our different reduced coenzymes are, the FADs and the NADs. If you prefer written versions of it, here it is. Okay, so our acetyl group, 2 carbons, released from coenzyme A and combines with our oxaloacetate, that 4-carbon compound, to form the 6-carbon citrate. First thing that happens, citrate is dehydrogenated and decarboxylated. That means we have a 5-carbon compound, and because we're going through those two processes, carbon dioxide and reduced NAD are also made. That 5-carbon compound undergoes the same processes, dehydrogenation, decarboxylation, to make that 4-carbon compound, plus the reduced NAD and carbon dioxide. That 4-carbon compound is then reorganized to provide the energy for ADP to be phosphorylated into that ATP that we saw. Now, that's the only point that we're actually generating ATP, remember. And this is an example of substrate level phosphorylation. So we're basically transferring that phosphate to our ADP to form the ATP. We then undergo a dehydrogenation for that 4-carbon compound so that we're going to generate reduced FAD. and that's the only point that we have the reduced FAD, remember, in Krebs. And then finally, our 4-carbon compound is dehydrogenated to form the oxaloacetate, our starting point if you like, and alongside that we get reduced NAD. It really is personal choice as to which version you prefer to learn. If you obviously like the idea of the diagram and then just memorising which bits go where, perfect, do that. Use the techniques where we're going to cover up with little flaps on the diagram and turn them over to check if we know them in the right order. If you prefer to go through and do the written versions, then do that. At the end of the day, revision is what works for you as an individual. There's no one right way to do this. You need to obviously identify the best way that's going to get this information in your heads. There are questions that come up on Krebs cycle where they're going to give you basically an outline of Krebs cycle with some of the parts on there and other bits missing. So the key thing that you need to be able to do there is clearly fill in the blanks. And that's where those carbon dioxides, reduced NADs, reduced FADs, ATP, etc., they're all going to be very important alongside citrate, oxaloacetate, and of course the acetyl-CoA coming in. Here's our little summary about what we generate at the end of Krebs cycle. So the end products, 6 reduced NAD, 2 reduced FAD, 2 ATP, and 4 carbon dioxide molecules. The reason that we get those numbers is because we have got 2 acetyl-CoA entering. That means we're going to go through Krebs cycle twice for each molecule of glucose we started respiration with. So just go careful. Obviously, for each acetyl-CoA that enters, we would have 3 reduced NAD, 1 reduced FAD, 1 ATP, and 2 carbon dioxides. But because we do Krebs cycle twice, because we started with 1 glucose and became 2 pyruvates, those 2 pyruvates then became 2 acetyl-CoAs, we go through it twice. So 6 reduced NAD, 2 reduced FAD, 2 ATP, and 4 carbon dioxide. One thing I am going to mention here is just a reminder that when we're talking about respiration, it's not a case that the only thing we can respire as organisms is glucose. Glucose is not the only substrate that is respired aerobically, even though all of the examples you look at in any of the questions that you've probably seen will be glucose. We just use that one because it's a nice clear pathway for you to be able to understand exactly what's happening start to finish, okay? But there are other respiratory substrates that organisms will utilise. So things like fatty acids, for example, will still be respired aerobically, but they're actually going to be entering as acetate in the Krebs cycle. If we're thinking about glycerol, that's going to be converted into pyruvate, so that means it's going to filter in at that stage. If we think about amino acids, these would undergo a process of deamination and then they enter Krebs cycle. So we can use these different respiratory substrates, it's just that they're not all starting at that very beginning point at the start of glycolysis. They basically filter in at different stages depending on what the substrate is, but they still go through aerobic respiration. So go careful not to think that it's only glucose that's ever respired aerobically in an organism, because there's lots of things we can utilise. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so that you can see when the next videos are uploaded, and of course head on over to the A-Level Biology website where you can find a range of other resources to help you in your A-Level biology studies.